here. And it's my pleasure to introduce the speaker of today. It's Gareth Difford, who is a researcher in NMBU in Norway, Norwegian University of Life Science, and he works in the breeding, genetics, and sustainable fruit production system group. He has established his own research team, the Phenotype Forge, where his group focuses on integrating phenomics and genomics across livestock and aquaculture species. Gareth did a joint PhD in Aarhus University in Denmark and Wageningen University in the Netherlands, where he developed a method for phenotyping methane emission and feed efficiency in dairy cattle. And in 2018, he joined Nofima as a researcher in aquaculture breeding and genetics before moving to NMBU in 2021 to begin his tenure-track position. Gareth is a strong advocate for Mike Coffey principle that phenotyping is king, and is passionate about adapting and developing new phenotyping methods to meet the challenge of modern breeding. And so today it will be around emerging phenotyping methods for feed efficiency and in Atlantic salmon. So thank you, Gareth. And so up to you, do you want to take questions as we go on, or do you want to take questions at the end?
I had planned for questions at the end.
Okay.
If that's all right. Right, keep me on time. I'll do my best as well. Yeah.
Very good. That's anything.
But really, thank you very much for the invite, and thank you very much for having me. It's my first time in Scotland, and it has not disappointed. It's been really fantastic to see a bit of Edinburgh and Roslin Institute. So I will tell you a story about phenotyping and feed efficiency in Atlantic salmon, but it's actually two parts, two stories. But before I get there, a little bit of shameless advertising. I'm one of the co-chairs for the aquaculture session of the WC Gulf, and it's 1 month until the deadline. So please, those of you who are very strong advocates for aquaculture genetics, please come join us and we can show the livestock people who is best. Most of this work is coming from the Research Council of Norway, two projects, the DigiFission project and Foods of Norway. And we have a collaboration with NMBU, Nofima, and Aquagen. But it's one of those things where the PI will try to present some of the students' work and not mess it up. But that's the most recent story. And actually, I share this slide because I will tell you some stories and show you some results of projects that have spanned the past 7 years, and there's a lot of people that have been involved, and friends, colleagues, and collaborators. So I acknowledge them here because I'm grateful, but also I'm always surprised at the amount of people skills that is needed with phenomics. Without further ado, we'll talk about feed efficiency. And in the case of Norway, more than 50% of the variable production cost of salmon production is the feed. But what a lot of people don't know is that actually the feed is indirectly responsible for 70 to 80% of the carbon footprint. And that is because of our over-reliance on Brazilian soya. But it's one of these stories where if you can produce more edible product for less feed, you can sell this very easily because it's more profit and also better for the environment. So it's one of these lovely win-win situations that's great for funding. But there's a catch, and that is that you need to know something about feed intake if you want to know something about feed efficiency. And with the Atlantic salmon, it is incredibly difficult to figure out how much each individual fish is eating a day when you have 200,000 of them in a net pen. So we need methods that are accurate, precise. We need high-throughput methods. So we need to be able to phenotype thousands to tens of thousands of individuals. And of course, it has to be heritable. And one thing that, one glitch that we have is that we don't feed individual fish the way you do with livestock. We feed the pen or the tank. And that actually brings me to the gold standard method of recording feed intake in fish. And that is the tank method. So you know how much you put in the tank, and you have some kind of collection device, and you know the mass of what you recover that's not eaten, and some kind of correction for water. And this tells you how much the tank ate. And it would be fantastic if we could put one fish in one tank and then we would know what's going on. But when you do this, they don't eat and they don't grow. So there's no feed intake, there's no feed efficiency. So what we do is we put families together. And so you have a family together and you know what they're eating, and they are pretty happy in this because they're sociable animals. And this method has some uses. So I mean, we know it's the most accurate and precise method we have, but it only accesses between family genetic variation, not within family. It is very costly. So Nofima has 70 tanks, so they can assess 35 families, and NMBU has 60, so we can do 30 families. So with pretty much all the tank resources in Norway, we can't even assess a normal 200 family breeding program. But there are methods that can do individual feed intake. And this one is an oldie, but it's a goodie. So what we do is we take tiny radio opaque glass feedlets and we put them inside the fish pellet. That's what you see here in step 1. We then weigh out known masses and we x-ray them, and we count these little pellets inside and we develop a calibration equation. It's then a matter of feeding your tank and x-raying your fish and counting and then inferring the feed intake. And this method was done quite extensively in the early 2000s in Finland. So what you see here is some rainbow trout from Antikausa, and they've been x-rayed, and inside the belly you can see these radiopaque beadlets. And it was lowly heritable, but it was heritable. And some have done this in Chinook salmon, but no one had done it in Atlantic salmon. And we thought, you know, X-ray has come a long way since the days where we had to hold up to the light. Now they have digital images. So we thought, let's have a look at this method. So I'm going to be telling you a bit about that. But it's not just the feed intake. We often focus on the problem of feed intake, but we want to transform these pellets into edible product. And so we need to know things like average daily gain, how much they grow, but we also want to know the fillet, so the edible part, and we want to know the composition. And it seems deceptively simple, bridging these two, but there's a whole whack of unknowns in between that we want to answer by phenotyping. Our colleagues that work with the Mediterranean species often laugh at us and say, you have 2 fish species with Atlantic salmon. And you see this in the way that we phenotype and the way that we breed, because we have freshwater par, and they go through a transformation called smoltification, and then we put them at sea. And maybe you can guess the answer to my question, when do we actually phenotype? And it's really reflected by this dichotomy in the biology. And that's that we only phenotype smolt and disease challenges right before they go to sea. And then after a variable amount of time, when they come out, we have growth and we have things like harvest traits. But when it comes to feed efficiency, the wheels fall off a bit. So we have 99% of the research is done with these tank level feed intake, and that's done in par, really small fish. And on the other side, we have our lipid traits, our harvest, which are measured at harvest when the fish are dead. So they're measured once, and that's at the end. So there's a complete disconnect between what goes in and what the product that's coming out. And why this is important is show some results from an old paper. It's a bit dated, but it was a mammoth research task. They took a cohort of fish, and at regular intervals, they would sacrifice, and then they would measure the composition. So from egg all the way right to the sexual maturation. And these 2 graphs show the lipid composition in the top and the protein. And if I show you the protein composition, it's fairly linear through their life. But if you have a look at the lipid composition, you see that there's an increase until this multiplication. Something drastic happens, and then there's a more drastic increase. And what this means is that every gram of average daily gain has a very different energy composition depending where you are along this curve. So we want to bridge this gap. We want to push these methods further so that we can phenotype feed intake and lipid utilization. And so I'll show you some results where we evaluate the feasibility and see how this has progressed. I will present results from a number of trials, but I'll describe this one first because they all follow very standard design. That is that we get something in the range of 30 to 35 families from the nucleus. In this case, it was from movie genetics. And the moment they're at eyed egg stage, so we know that they're fertile, we put them all together. And once they've grown to the size that we can pit tag them, the radio frequency identification tag, and we can take a tissue sample for genotyping, then we can split them. And in this case, we had a gold standard tank trial with 1,700 fish, and we had an X-ray trial of 700 fish. And these two ran in parallel at Mephima's Sundalsura. So we had feed intake every single day for the families, and we had each family was in 2 tanks, so we had a replicate. And then at the same time, we had their siblings in 2 big tanks, and we would then feed them these radiopaque beads and X-ray them at regular intervals. And this was very lucky funding from 2 EU projects, Aqua Impact and New Tech Aqua, that were running at the same time. Without that, we wouldn't have been able to do this trial. So I will talk you through this busy slide. But I absolutely love this slide because it is the process that is involved in getting the phenotypes. And it's really, it's like this interaction between what physical infrastructure you have, your human resources, your understanding of the species biology, getting it all to work together. So in this case, we have our families, about 700 fish, and they are the day before, their normal feed is swapped for the feed that has the glass beadlets. We then pull the plug on the tank and we transfer them 50 meters down the road to a building that's lead shielded and it has the X-ray. And then it's a bit of a human pipeline. So this man here anesthetizes fish in batches of 6. We read the pit tag and the length and the weight of each fish. And they then go to this mammography X-ray. So it's designed for human purposes, but it's very good at looking at fine tissue like we have in Atlantic salmon. So they are X-rayed, and this lady with the lovely floral curtains, she develops the image and links the ID. So, and then we get this X-ray and we return the fish to a highly oxygenated tank, and they recover from anesthesia. So this whole process is designed to minimize their time out of water, and it's something like 94 seconds from tank to tank. So this guy's job is very important. He controls the anesthesia and the flow. And this lady's job is also very important because without her, we lose the ID and then we can't do anything. So we did this in the first trial. We did this 2,100 times and we only lost 6 fish. So we were very happy with how this machinery works. And I kept thinking to myself, why are people not doing this more often? And then this happened. Then the images were there, hundreds of images, thousands of fish, hundreds of thousands of beads. It was COVID, it was winter in Norway. Somebody had to count these beads. That somebody turned out to be me. So I went down the route of classical image analysis, and I had some very nice collaborators who wrote this into a program so we could read in the fish, mark the region of interest, and then we did some filtering and background correction, and ultimately it would count and it would show you what has been counted. So we could always correct, if two beads are touching and or if it's missed something. So there was a little bit of manual annotation on top, but this suddenly made it bearable. So what I show you here is as the fish get bigger and we have more beads, how long it takes me to count how many are in there. So the orange is me doing it manually, and that doesn't include the times I make a mistake, have to start again or give up, walk away, think about how I ended up here in my life. But the underneath is with the software. So it still takes time, but a lot less. And when it's COVID and when you have a toddler and you have to do this, any type of time saving makes you smile. So we had our data now to have a look and see, is this working and how does it compare to the gold standard tank level feeding? And actually, it's quite a simple model. It's a bivariate model. We had a few fixed effects and we fit this random effective family. You can fit it as an identity matrix, you can fit it with a pedigree, you can fit it with a special G matrix. It gave very robust results across. And actually what I show here is the ranking that we get from our families across this. And really, when you think that we have 70 observations, 35 families, it's fantastic to see this strong linear relationship, and the means were very similar and the variance components. So this was the first sigh of relief. Okay, this could be working. But we could, with the X-ray, you have individual level, so you can now do individual level variance components. So I just plot here the breeding values for feed intake on the y-axis and average daily gain on the x-axis. They were significantly heritable, and we could find variation at any fixed point on the y or x. So we were very happy because this shows me there could be feed efficiency. But the fish get bigger. They don't wait for us, and the number of beads increases. There are ways around this. You can get smart by changing the feed and changing the how it's diluted. But now the classic image analysis had no chance. Now it was completely. We had lots of shadowing where they reflect onto each other. So when you are faced with a challenge that's insurmountable, get a PhD student and make it their problem. So Akil Ahmad, he is defending November, and he took all of our images and we retrained a YOLO model to now find these beads. And so he got it, and it will here every single bead is marked, and we have a confidence on each bead. He validated against the manual counts, and we had R-squares of 99, so we were happy. And he's shared this in his GitHub, so it's freely available now. Now nobody else has to do this ever again. But if there's one thing, if there's one legacy I leave, it's that nobody else has to count. And I can refer you to his paper. He's got all the genetic parameters. But there was one problem with this, and that was that we sedate these fish quite heavily so that they don't move while they're X-rayed. And Atlantic salmon don't enjoy this so much. So it takes them about 8 days to start eating again. And then, you can't measure them straight away because there's been very little growth. So there's a limitation on how often you can do this. So we said, okay, let's try something new. So we scratched the glass beads. We changed it for iron beads. And swapped out the X-ray for a metal detector. And so now this is now one of our more recent projects with Aquagen. And we have a lot bigger fish. So this time we grew them to 2 kilos. And so you present the fish on a conveyor belt, and it goes through this detector, and it gives you back an intensity reading that we relate to the feed intake. So we got heritability estimates in the big fish, which we were happy about. And this is the work of another PhD student, Nicholas Jacob. But we're having problems with the small fish. So we are not sure if this works in small fish. But what's really nice is they start eating almost 48 hours later. So it's a lot more gentle on the fish. And we've managed to do over 1,000 in a week. So it's quite high throughput. So that's the feed intake part. But we also have this lipid problem. So we want to know the composition of what's growing, not just what the wet weight is. And perhaps some of you know, but there's a whole lot of destructive lab methods for extracting lipid. So something like a phos or a Blyton dye. These are, you have to grind the samples and lyse them in acid and then get them in solvent. And I did this recently, and it's about 80 pounds a sample. So it's quite expensive. We had a project where we scanned in the broodstock, so in big fish, 5 kilo, the fillets with a near scanner. And we were able to phenotype with both methods on 523 fish from benchmark genetics broodstock. And because no one had validated this near scanner, there were some questions about whether it worked. So we had a look at the genetic parameters, and we got this genetic correlation of 0.91. So we were thinking, okay, well, that's fairly decent relationship between these two methods. So the high throughput method is we can trust it to some extent. And there's this question, and it's been done in a few species where they homogenize the fillet and then do a near. So you don't have this spatial discrepancy. So you take the whole fillet, blend it, and it makes these horrible looking brown homogenate. And after doing that, we only managed to push this genetic correlation up 2 units to 0.93. So it's kind of a trade-off. How much work do you want to do on the phenotype to try and maximize this parameter? But it wasn't a completely wasted effort, because the near that we had here gives us individual absorbance across the spectra. So we could have a look at the individual wavelengths, and we could see if they were heritable. And this you might have seen a few times before in things like milk. And there were regions that we know are associated with fat, and they were highly heritable. And we could do the genetic correlations to the gold standard, this lipid extraction. And there were regions which are extremely highly correlated. So we knew we don't actually need this whole spectra. There are windows that we can use. And this next method it's a bit of a story, because I wasn't here for all of it. But I tell this story because I think it's a very nice example on how technology is not static, continually develops and changes. And a good example is this interactance near. So most things that live on our planet under the sun have a protective layer of skin that stops light from penetrating. It protects our cells. And with Atlantic salmon, they have the scales and the skin, and it is very hard to get a measurement through the skin. So some researchers from NAFEMA and I believe Syntef developed this probe called the Q point. So you touch the surface of the skin, and what's happening here is the light goes in at different angles and bounces up the probe. So it's not transmission, it's not reflectance, it's something different called interactance. And so they validated this method, and it worked really well. And then they decided to make a commercial version called Q-Vision. And I haven't used this myself, but I wish it was around when I was applying for projects, because it fits in a suitcase, and you pass the sample over the probe, and now you're getting a near interactance reading for things like fat. Now, for whatever reason, I believe nobody bought this, and so it's not available anymore to use, even though you can do this on live fish and you can get through the skin. So it's a bit of a pity that it didn't go any further. But they didn't give up, so they made a research version, and then so the next development was you don't have to touch the fish. Now we are doing it with incident light. And so there's different bands of light here, and they penetrate at different depths. So you get a better reading through the fish. And so for Akil's PhD, we measured in our movie fish that I spoke about earlier. And we got these spectral measurements. And they are a subset. So we don't have the full wavelengths now. We've got the important ones. And now we also have different depths. So we've got new information through the skin, live fish, different depths. And we had a look at this. And I show the heritabilities for whole body fat on the diagonal. And on the off diagonal, I show the genetic correlations. So in the early life phases, it's whole body fat is very highly correlated. But when we get to later life phases, this correlation drops. And this correlation is important because it matters when you do your feed efficiency trials. Are you doing it on the big fish, or are you doing it on the small fish in freshwater? But there was one caveat here, and that is that what was done in the green box was this interactance near. Outside of the green box was done with another method called the distal fat meter, and that was because of COVID. We couldn't get to the fish when they were bigger. So there's a question mark here. Is this some type of life stage G by E, or is this because of a different method? So we now, in the most recent project, got permission to do this in big fish and live fish, and I can show you a little video here. Focus down here. And you see it just takes a few seconds to acquire the spectra, and that's how quick it is to do in a large salmon. And if I can do this, anybody can do this. Now we have a new near, now generation 3, but the people who developed it were burned on generation 2. But I'm happy to say that Sintef and Nofima have now made a generation 3 called the Sense Inside. So it's this, now the light is this cool looking circle here. And the box is about the size of an Xbox. If you don't know what an Xbox is, it's like a phone book. So it's highly portable. And it's semi-commercial. So what I mean is they are considering commercializing, and now they're in testing phase. So it's, and I hope they proceed, because this is super useful for us with the lipid studies. So what I hope I've impressed upon you with these stories is that the phenotyping, you always need to push these methods a step further, every single time, a step further. Try get them to be more flexible for your species so that you can access the information and understand the genetic variation. What I showed you is often small and incremental steps, but they are super necessary if you ever want to get a phenotype from lab to actually into a selection program. And I hope one day in my career I'll actually achieve this, because it hasn't happened to date. We test at every single level, phenotypically and genetically. And what has been really encouraging is that at every single step we've found untapped genetic variation. So with that, I would just like to thank you for listening, and hope you have some questions for me.
So for the near or the new methods that you're using, are you always doing it at the same place in the fillet? And have you seen differences in different portions of the fillet, or is it quite consistent what you get? Is it?
Yeah, no, so this type of standardization is super important. So we have this cut that's often used for testing the Norwegian quality cut. So we put it in the same position, which is pretty much the top middle of the fish. And we take from both sides. So we take a replicate. This is not for breeding, but the people who do this for on the slaughter line for sorting for quality, what they do is they have a little arm that orients the fish head first, and they take they get their acquisition along from the head all the way to the tail. Yeah, so there's some flexibility in how you can use it.
You have multiple measurements on the same fish, and then you do like an average or something.
Yeah, yeah.
Do you have to calibrate it? So you would do also another method, and then you calibrate your measurements to predict the value.
Every single time.
Yeah.
So it's not a standalone method. So you have to take some of your samples. Usually we take something like 50 or 40 and send them for the reference analysis. And then you retrain or update your model. And we use partial least squares regression models. We try many different type of machine learning models. They never ever beat the PLSR. So it's always a competition to see, can a new method beat PLSR?
Interesting.
Yeah.
I was using a flat emitter in France for random terms. And it felt like it was just random.
The distal fat meter.
Yeah.
Yeah. So that's quite an interesting tool because it feels like a random number generator.
Yeah.
But with enough replicate measurements, you do get to, yeah.
And it just runs the feed intake. So because it relies that you have to put something what is it again you put in the pellets?
It's little glass beads.
Yeah. Because what is the repeatability of that, right? Is that because you only measure it once, right? Because it will be quite expensive. Or do you have repeatable measures of feed intake, and how repeatable are they?
Yes. So we try to do at least 3 measurements,
Okay.
but for a fixed growth period.
Mhm.
So we did, I think it was from 50 gram to 300 gram, which is quite a big growth period in the small fish. Now I can't remember the repeatability estimate, but we were bragging that it was 2 or 3 times higher than the previous times people have done it with old X-rays. So yeah.
Because is that a bottleneck? If you now did that on a routine basis, or if breeding companies took it on,
yeah,
and you were breeding for the deficiency or feed intake to get it to account, is that the glass beads? Is it expensive, or is that something you can do for routine?
Yeah. So I had a lot of fun with these glass beads because if you read the old papers, there was a company in the UK actually that sold these beads, and that company was bought up by many other companies, and I couldn't trace what happened to them. But I found a company in Germany that makes these beads, and they have many different properties, different ways of mixing the glass. And what they use it for is if you want to take varnish off of furniture, you can sandblast. So you can shoot these beads at high pressure to take off. So they have different sizes to how gentle you need to be. So I got them to send me a sample of all their products, and then we X-rayed them. And well, actually, they all showed up on the X-ray, but I was hoping to narrow down the range. But yeah, so they're readily available. They're quite cheap as well.
And they're not. There's no harm on the fish. Is that proven? Is it an issue that could be harmful?
So we did a bit of work on this because when people used to use this method, they used a pelleting press to make the feed. And since then, the industry's moved to extruders, which are high pressure, high temperature. And there was some of our collaborators were concerned the beads would smash or break. So we had to do a bit of testing, making feed and then X-raying, having a look. And we did find some beads that were cracked. So they do take the pressure. But no scary looking glass shards or anything. And we haven't found any inflammation in the distal intestine. So we think that they are fairly innocuous to the fish. They just pass through. Let's go.
Just to follow on this, could that be used in commercial farms? Because when they go into the food chain, I suppose that there is the need to have some approvals, something like that, to use those crystal bricks.
So they, what happens is they pass through the fish, and we had to do some pilot studies to figure out how fast do they pass through the fish. And it takes something like 2 days to completely lose them. And what usually happens in the industry is before harvest, they fast the fish, sometimes up to 10 days, but normally a week. So we're pretty confident that the digestive tract is completely empty. But we've never done this on the really big fish in the commercial setting. FEMA has invested in a very nice portable X-ray unit that can fish. So the last test I did before I changed to the university, we had a 6-kilo salmon and we could fit it diagonally on this portable X-ray. And then since then, we haven't had any luck with calls or funding for testing on big fish. So we haven't reached the point where I can answer your question yet. Yeah.
Your image in the bottom right there, like some kind of tank or independent solution. Can you see that becoming an option? In terms of maybe the glass beads, maybe the iron, maybe something else, in terms of leaving it independent, having a pit tank and corresponding value for each fish that swims through.
So this is another story that wasn't told today. So this is a diode frame with a pit tag reader, and then just used for length and predicted weight. But were you asking me, do I anticipate something like that for feed intake? Or were you asking how?
With speeds, but feeding
Yeah.
Some of the metric.
You know what I would really love, and I'm not sure if we'll ever get there, but this X-ray, this metal detector that we use, which is out of water metal detection. There are companies that have metal detectors that work with pipes, and this is to stop sort of factory metal getting into your milk or your yogurt. So I don't know if we're ever going to get a chance to get high-risk, wacky funding for something like this, but it would be cool to have this pipe solution. And then the trick is how to make the fish swim through the pipe when you want it to swim through the pipe. So there's... But,
maybe when you transfer them.
Yeah, or maybe when you transfer them, yeah. But so how you are thinking now, that is the heart of phenomics. It's how to.
Wondering how much variation there was in feed efficiency in wild fish. Of trying to get to an understanding of what we've actually done when we've domesticated these guys, how much indirect selection or direct there has been on feed efficiency in just making fish grow faster. So we've done any of the stuff on wild fish, how variable feed efficiency is and how relatively variable or canalized it is in the domesticated.
No, not at all. Not at all. We haven't tested it. We haven't looked at anything like that, and it would be.
It's highly variable as a trait in the farm population still, which have been selected for growth.
The one thing with the feed efficiency traits is it depends how you define them to say how variable. So in one of the Keel's papers that I referred to, which was in fish up to smoltification, when we use this trait definition, genetic residual feed intake, the heritability is 10%. And when we use, I'm not sure if you're familiar with these, but phenotypic residual feed intake, it's a little bit higher. But the aquaculture industry is kind of hooked on this metric, feed conversion ratio. and that one is at about 19%. So we still have heritable variation in these different feed efficiency metrics, at least in the early life phase. Yeah. Andrea, do you want to?
Yeah. Go beyond your study because you know so much about the phenotyping. So in domestic livestock, a lot of these phenotyping methods go in tracking individual animals, so in identifying individuals. How far in aquaculture? I think we had a little bit discussion. Can just summarize. Is that also a direction that the whole aquaculture industry is going? For example, study feeding behavior, going on to the behavioral. What is the state of the art in that?
Yeah, so it's quite interesting. I think the field is wide open. That doesn't mean people haven't done a lot of work in it. So one example is if we are talking about short-term video, where you can see every individual in the tank. There are tracking methods where we can just label each fish and say this is fish 1, 2, 3, 4, 5, and you can track them and their behaviors through, and that's reliable. So that works short-term. And then there's been a lot of work coming out of Czechia, a group in Czechia, and another group in New Zealand. They're doing different kinds of landmarking or fingerprinting. So taking things like spots or the shape of the lateral line and using that to re-identify with photographs. And what you see, it's always in laboratory scale. And they've got re-identification rates sometimes 98%. So it looks really promising.
Mm-hmm.
So I feel like, without having done this myself, I feel like the technology and the models are actually there, or they're pretty close. But now they have to expand the scale. So we need to measure them for a longer period of time. You need to have more fish in the tank, or as you get more towards commercial kind of setting, or you have them re-identified or their identity confirmed less and less frequently. So it's the answer I'm giving you kind of fits in with my take-home message that there's things that work, and then you need to push them further and further and further until we get something that's working on commercial scale for breeding.
So when you say the research scale, how many fish can you track at the moment?
Yeah.
Are we talking hundreds or thousands?
When I last looked at this field, it was something like 125 to 250. Yeah. And we're not quite there. But honestly, it's an exciting space. So there's a lot of companies working on underwater camera technology. So I wouldn't be surprised if there's developments. Pleasure. I am just trying to check if there's questions online, but I can't see on the screen. No. Okay.
And one last question on the room. So the same Gareth Difford presentation.